Thanks for the very nice introduction. Share my slide here. Good morning, everyone. My name is Rose Zhu. Today I'm going to talk about the precision gene silencing to support the integrated pest and pollinator management. Sorry. Here we go. In our first lecture, Dr. Rosot introduced some very basic concepts of IPM and IPPM. In generally, the IPM is try to use multiple tactics to for the crop protection in a way that can minimize the minimize the ecologic, health, and environmental risk. The IPPM incorporate pollinators into the system by consider the pollinator protection as a key component in IPPM. In this new system, how to balance the pollinator protection and the pest control is very critical. Therefore, the new genomic technologies that Christina and I will focus on today hold a great potential to support this balancing act. Because these genomic technologies not only can manipulate the pollinators and the pests themselves, but also they can incorporate, they can manipulate the host plant resistance, discover new pesticides, and also boost the biological control. As the definition of genomic technologies, they are technologies used to manipulate and analyze the genomic information, such like the gene silencing technologies, sterile insect technique, omics toolkit, as well as the macromolecule structural analysis by the X-ray crystallography and MR cryo-EM technologies. By using these technologies, we can understand the fundamental rules of life, which can indirectly support the IPPM. In addition, these technologies can also serve as tools to directly control the pest and protect pollinators. Today, we're going to focus on the newer development RNA and CRISPR-mediated gene silencing technologies. Both of these technologies actually share many similarities. The first, both of them are state-of-the-art technologies that win the Nobel Prize. For example, the RNA interference won the prize in physiology or medicine in 2006, and the CRISPR-Cas9 won the chemistry prize in this year. The other similarity is both these technologies actually is a natural process for the organism who can involved to defense against the virus attack, which are highly associated with the coronavirus outbreak, what we are fighting now. For example, the RNA is a natural cellular process, a defense against the virus attack in plants, insects, nematodes, and other animals. Once the virus infects the organism cell, the double-stranded RNA of the virus will trigger the antivirus defense pathway in the organism, which will use this double-stranded RNA of virus and target and eventually cleavage the virus RNA in the organism. Similarly, the CRISPR-Cas9, which is the observation of a clustered, regularly interspaced short palindromic repeats. It is a family of DNA sequences found in the genome of most bacteria and archaea. And this CRISPR is thought to derived from the DNA fragment of the virus that had previously infected the cells. For example, this new species derived from the virus attacked, they will incorporate into the CRISPR sequence with this new species in the cluster, and they were separated by the repeat, similar sequence of the repeats. Then this species and repeats will transcription to the CRISPR RNA, and then they're going to incorporate in the molecular machinery, such like a Cas9 protein, to target and destroy the genome of a virus. Both RNA and CRISPR technologies, they are very effective in most many insect species. And also they can they can provide the precise gene-specific silencing. Yeah, for example, there are many pathways, RNAi pathways that have been identified in the insects. The small interference RNA pathway is which causes the silencing, gene silencing. For example, you can design a double-stranded RNA based on the specific gene sequencing that you are interested. Then you introduce this double-stranded RNA of your target gene into the organism. Then the dicer protein in the organism will chop this long length double-stranded RNA into short, small interference RNA, we call the sRNA, which is only 19 to 21 base pairs. And then this sRNA incorporate with the RNA-induced silencing complex proteins. They will use the one strand of this sRNA as a guide and find their complementary sequence on the target mRNA. Eventually, they cleave this mRNA and cause this gene silenced. The other RNA pathway identified in the insects are mRNA. Yeah, typically, this mRNA is a non-coding RNA, single-strand non-coding RNA, and they usually is triggered the regulate the gene expression. The CRISPR-Cas9 is involved in a single guide RNA and the Cas9 protein. The single guide RNA, which including 20 nucleotide target-specific CRISPR RNA sequence, which we designed based on our target gene sequence. And then followed a PAM. PAM site is a G, usually is a NGG. Then followed by a trace RNA, which will bind on the Cas protein, Cas9 protein. When the single guide RNA and the Cas9 complex bind on the target site, the Cas9 will introduce a blunt ended and double-stranded cut 3 bases upstream of this palm site. Yeah, after you have this break, they will either undergo the non-homologous end joining, which will replace this break size with the insertion or deletion. Yeah, in this pathway usually can cause the knockout of the gene. In another way, they can undergo the homology-directed repair, which is required specific sequence, which will be insert and replace the trapped site of this gene. For example, if you can use this method to replace a resistant mutation in a insect pest with the wild type sequence. Both IR and CRISPR-Cas9, they can be used as a loss of function tool to facilitate the functional studies, help us to understand the functions of genes. For example, in a tribolium, which in the tribolium parathird resistant strain, QTC279 strain, we hypothesize cytochrome P450 genes may contribute to the parathird resistance. So we introduce the double-strand of this P450 and silence this gene in the resistant strain. Then we perform the bioassay with the deltamethrin, which this strain resisted to. After knockdown this cytochrome P450, the resistance ratio decreased dramatically from 4,000-fold to 44-fold, suggesting this cytochrome P450 may play the role in the deltamethrin resistance in tribolium. Similarly, the CRISPR-Cas9 can also used as the functional studies tool. For example, in this study, the scientists use the two single guide RNAs and the Cas9 protein introduced into the eggs of cotton bollworm. This species actually, the RNA doesn't work very effectively in this species. So after you trap out this entire P450 cluster from the genome, you perform the bioassay with several plant allochemicals and insecticides. They found after knockdown this gene cluster, the susceptibility to the some plant allochemical and insecticides enhanced significantly. Suggest this cluster P450 cluster play roles in the chemical adaptation in these insects. We have to the RNA and the CRISPR-Cas9 is very effective for these research studies. However, they only can serve as the tool. They only can provide one evidence for the function of these genes. In order to understand the function of these genes, you have to perform you have to provide other evidence. There are many delivery methods have been developed for the RNA. And there are also some method can be used for the CRISPR-Cas9 delivery. One method we call the injection. We can perform the marker injection for the double-strand RNA. You can use a marker injection, introduce the double-strand RNA in all stages of insects. But for CRISPR-Cas9, you only can inject the eggs. Also, another very economic friendly method we call the aspirator injection. You can use the for RNA, introduce double-strand RNA to the insects in all stages. Also, recent new method for the CRISPR-Cas9 delivery, we call the REMOT control. They can use this method to introduce the single guide RNA and Cas9 into the female adults. I think Christina will talk about this method for more details later. The other main method is feeding of double-strand RNA. This one most being used for the RNA interference. This double-strand RNA can be chemical synthesized or can be generated using bacteria, genetic modified bacteria strains. And also you can transgenic express the RNA into the crops. And this transgenic plants, actually the CRISPR-Cas9 also can be used as a one method. So because this feeding delivery method can introduce a lot and consistently introduce this double-strand RNA into the system, they hold a great potential for the IPPM. They can be used in the field, right? So that's have a great potential for the IPPM integrate pest and pollinator management. So in the following slides, I'm gonna talk a little bit more detail for the bacteria synthesis double-strand RNA and transgenic plant. For the bacteria-mediated double-strand RNA synthesis, generally is you construct a plasmid by insert your target PCR product into a L4440 vector with 2 T7 promoters on the two sides of the insertion. After the construction, you can transmit this plasmid into RNase3 deficient E. coli strain. When you use IPTG to introduce this E. coli, excuse me, this bacteria will, excuse me, automatically synthesize the double-strand RNA in the cells. So by using this way, you can, automatically synthesize a lot of double-strand RNA. Currently, this method can be used, in the lab for the functional genomic study. For example, you can screen the target genes in the lab with check their phenotype by, perform the phenotype checking. Once you selected some important, effective target genes, which can either lethal manipulate the pest, or you can change their reproduction or change their, physiology development. Then you can use them as a target in the field, in the greenhouse, or eventually in the field. Once you found some, resistant development in the field, you can come back to the lab, simply change this target gene, or you can introduce the multiple target genes together. This method actually, the advantage of this method is the dramatically decrease the cost of double-strand RNA synthesis from $12,000 to only $2 for each 1 gram of double-strand RNA. Yeah, therefore, currently, many companies adopt this method, try to commercialize them as RNA-based pesticides. The another delivery method is we directly genetically modify the crops. Actually, actually, everything we eat currently have been modified genetically through domestication from the wild species and many generations of selection by human for some desirable trait. However, this natural happening process may take hundreds to thousands of years. With the new genetic technologies, you can, with purpose specifically modify the genes, and then which can dramatically speed up this selection process. Yeah, currently the delivery method for the transgenic modified crops, including the Genomic method or soil bacteria, agrobacteria, which is the cause agent of crown gel disease. And of course, very recently, we use a CRISPR-Cas9. We can incorporate this new technology with a gene gun or agrobacteria delivery method to generate the new, more fast and effective CRISPR-Cas9 transgenic crops. For the CRISPR-Cas9, they also have other delivery method, beyond these two basic transgenic techniques. Some of these methods, for example, the PEG-mediated CRISPR-Cas9 vector delivery, because they introduced very few DNA genes into the organism, they probably without the issue of regularly, regular berries. We need no worry about the regular berries. These slides show you the trees or development objectives we can consider when we genetically modify these crops. These trees, including the insect resistance, called RI, such like the BT crops or RI corns. Yeah, they typically is resistant to the pest. Or the virus resistant, herbicide tolerance, stress tolerance, like drought tolerance, nutritional enhancement product, or you can improve the product quality or increase in the yield or plant-based pharmaceuticals. These slides actually show you the current commercialized crops in US. As we can see, they are in the cotton, they're about 96% cottons. They are modified towards the insect resistant and herbicide tolerance. Since this figure is generated in 2014, I believe they're not in counting the RI crops and CRISPR-Cas9 technologies. However, in 2017, the first RNI corn was commercially released. This strain of corn called Smart Stacks PRO. Generally, this genetic modified corn can expressing 3 BT genes with 1 double-strand RNA. Yeah, the target of this strains of this corn is to control the Western corn rootworm. As we know, the transgenic crops, they really hold an advantage because they can precisely gene-specific silencing and protect our crops. However, the off-target effect or non-target effects of the RNI and the CRISPR-Cas9 can occur. Yeah, therefore, the non-target effects on the beneficial organisms should be carefully evaluated. Yeah, for example, the some RI crops. They can, this is the RI crops, RI corn. They synthesize the WRI of to target their the pest, but at the same time, you have to evaluate if this WRI have any negative effect on the like pollinators or biological agents or the other beneficial organism in the soil. Since this mode of action of the RI is based on the sequence of the species, right? Yeah. However, currently there are many species we haven't identified into in our ecosystem, and also many of them's genome sequence are not available. That's why this risk assessment is very difficult to cover all these species, in the environment. Yeah, that's one of the challenging of the um, RI crops currently, and also is a important point we should consider when we commercially use these technologies in the field. I think that's all for my talk. Any questions? Thank you.
Thank you.
Great, thank you, Rose. And I think we'll have time to take some questions at the end as well. So let me see. All right, are you guys seeing my screen? Just my one screen? Okay. All right. So thank you, Rose, for giving that background in terms of how these genomic technologies work and how they can be applied to pest systems. And I'm going to talk a little bit about how they could be applied to honeybees and other bees to improve bee health. And so I've sort of structured this that we're going to talk about the biology of the honeybees and talk about sort of their complex lives and the different characteristics and traits that they have, and how these traits actually create some challenges for trying to apply these genomic manipulation technologies to bees. Then we're going to talk sort of about a case study of, if we could we make bees that have sort of ramped up immune systems and therefore would be more resistant to different pathogens and parasites, and is that something that we would want to do? And then I'm going to end talking about some of the broader challenges sort of at the ecological scale in terms of applying these technologies to improve bee health. So I think most of the students in this course have not had a lot of background in bees, so I'm going to give you some of this basic background. So honeybees have three castes. There's workers, which are female, the queen, which is female, and then the drones, which are male. And they have a sex determination system that's based on haplodiploidy systems. So the drones are produced from unfertilized eggs, and the females are produced from fertilized eggs. So the drones are haploid, they have one set of chromosomes. The females are diploid, they have two sets. And whether or not you become a male or a female is determined by the complementary sex determiner gene. And this gene has multiple variants, so multiple sequence variants, which are called alleles, in a population. And if a bee has two different variants, she becomes a female. If a bee has a single variant, it becomes a male, right? And so a haploid egg has a single variant, so it becomes a male. But if you happen to get two of the same alleles because you're from an inbred population, then you're diploid, but you have a single variant, so you become a male. And as you'll see, bees don't really like things that are different, and so they can detect that there's these diploid male larvae in the colony and they eat them. And so those don't grow up into any sort of bee that supports the colony, and instead they get removed, and so that can be really detrimental for the colony. A lot of this work on complementary sex determiner genes was done by Martin Bai, who's at University of Düsseldorf, and I really encourage you. It's some really beautiful work that he's done over the years to tease apart this system. So what this means then for trying to sort of create lines of bees that have particular traits, you have to have a genetically diverse population that you're working with, right? So normally when you're sort of thinking about like a crop plant, you can have pretty much a single genetic line, that that has the traits that you want, and then you can breed from that. But in the case of honeybees, that's not going to work out very well because you need to have these genetically diverse populations so that you avoid getting these diploid drones. There's also a reproductive division of labor in colonies. So the queen is the only one who can mate and lay fertilized eggs, so lay female eggs. The workers are facultatively sterile, so they can't mate. They can activate their ovaries and lay eggs, which are unfertilized, so they become males. And they'll do this if the queen is lost or there is no brood. So basically, if the colony is in kind of a hopeless situation where they can't replace the queen, then the workers might activate their ovaries and produce drones. Sort of a fun fact, there is a subspecies of honeybees, Apis mellifera capensis, which is found in the southern tip of South Africa. And these workers can actually lay diploid eggs. So they are capable of laying diploid eggs, but the other species or subspecies, as far as we know, cannot do this. So whether or not you become a queen versus a worker is determined by the nutrition that you get as a larva. So until the larvae are about 3 days old, they can become queens or workers. They're considered totipotent, but after that, their development is fixed. And queen destined larvae are fed royal jelly, and worker destined larvae are fed worker jelly and some pollen. So they're basically given different nutrients, and this triggers these different developmental phenotypes. And so how does this become a problem if you're trying to, again, develop a line of bees that has some particular genetic trait that you've engineered? So this means that when we're trying to make these mutant lines, you have to do this by rearing these reproductives. So the queens and the drones have sort of special rearing conditions that they need, and you have to sort of produce them in a series in order to breed them together to get this line that you want. So for example, as Rose was saying, you have to inject eggs or embryos to get a queen and then rear this up as a queen that hopefully then lays haploid male eggs that has this mutation that you're interested in incorporated, and then you would mate this male with another queen, and hopefully you would get then a hybrid that would be heterozygous for this mutant and wild type allele. You'd mate this one to a male that has a mutant allele, and then hopefully start getting offspring that have the mutation sort of fixed in them. And so this again requires sort of a series of steps and stages and special rearing. Most of these approaches use egg injection, and as we talked about before, trying to get a colony to accept eggs or larvae that have been manipulated can be challenging because they can detect that they're different and they eat them. And there is in vitro rearing that's possible, but often these bees are not as healthy and can't really be used to start their own colony. So as Rose mentioned, there is an alternative approach that's being developed to egg injection, and this is called this remote control or receptor-mediated ovary transduction of cargo. And basically what happens here is that the CRISPR-Cas9 has a peptide added to it that allows it to be taken up into developing oocytes. So instead of kind of injecting into the egg and then hoping that it gets incorporated into the developing genome and gets into the germplasm, in this case you inject the mother, and then the carrier peptide helps incorporate the Cas9 into the developing eggs, and then those eggs should get modified at kind of a very early stage, right? So this should be a sort of faster way of creating these mutant lines. And this remote control system has been developed by Jason Rasgon, who's a professor at Penn State. And he's been collaborating with lots of people on getting this to work in different systems. So it's been used in mosquitoes and whiteflies and beetles and Nozonia. And so it seems to be pretty robust to working in a lot of different species. We are trying it with bees, but because again, of some of the challenges with the biology of these bees and getting enough eggs produced, we have so far not gotten any mutant offspring from this, but this is something that we're working on. So this remote system can overcome some of the challenges, right? So you don't have to do these egg injections. You can inject right into the queen. You can potentially skip some of these breeding steps, but it's still challenging. So the efficiency rate can still be pretty low. So the uptake into the eggs and the modification of the offspring is still quite low. So you can avoid some of the steps, like you don't have to worry about the microinjection equipment, but you still end up having to screen a lot of offspring to get the ones that you want. And then again, there's these other sort of challenges. So the purification of this protein can be tricky. You have to time the injection so that the eggs are being produced and vitellogenesis is happening. Often you have to do multiple injections. Concentrations are really important for getting it incorporated into the eggs. And then you have to include some other things like once it's taken up into the egg, it has to escape the endosome. So there's some other reagents that are included. And then of course for honeybees, we have to have some way to contain this. And since bees, need to be in full colonies in the field, this can be challenging. Another aspect of bees, which is really cool, but makes it difficult for applying these genomic technologies, is their mating behavior. So queens and drones mate about a mile or so away from their natal colonies. They fly in the air and then they mate in these mating swarms, and a queen will typically mate with about 12 drones. And this genetic diversity is not only important to make sure that she has lots of different alleles of that sex determiner gene, but also the genetic diversity in the colony resulting from a multiply mated queen is really important for colony health and productivity. So the more genetic diversity that you have, the less diseases you have, and the better the colony performs. And so you can instrumentally inseminate queens to maintain your stock, right? So it is possible to control the mating, but again, you have to have multiple genetic lines of bees available to do this breeding, and you have to do it continuously. And so this makes it challenging, for a beekeeper to be able to do this themselves. And so that means they're always probably going to have to buy stock from some genetically modified stock from a commercial breeder. The other thing that bees do is a worker division of labor. So these female workers that are in the colony, there's about 50,000 of them in a normal colony, and they perform different tasks as they age. So the young workers, which are about a week old, they work as nurse bees, where they feed the larvae with special food that they develop in their glands and their hyperpharyngeal glands in their heads. And then when the larvae are old enough to pupate, they put a wax cap over them. Middle-aged bees, so there's about 2-week-old bees, will do other in-hive tasks. So they build the honeycomb, they store the nectar, they do undertaking, which is removing dead bees from the colony, and they guard the colony entrance. And the oldest bees in the colony, which is about when they're 3 weeks old in the summer, are foragers, and they leave the colony to collect nectar, pollen, water, and propolis, which is tree sap, and they use this for blocking holes and also seem to have antimicrobial properties. So this behavioral maturation process is based on age, right? So with young, middle age, and older. But the rate of maturation and the direction is modified by the physiology of the bee and the social context of the colony. So if you take all the forager bees out of a colony, either as an experimental system or because they've been taken out by a pesticide or a disease, some of the young bees will become foragers when they're much younger. So even when they're just 5 days old, they can start foraging. But these precocious foragers are not as good at collecting food, and they don't live as long. So they don't sort of bring in as many resources to the colony as a normal forager would. Also, if you take all the nurse bees out of the colony, some of the foragers will revert back to performing nursing behavior. So again, this is a very flexible system. And so when we think about applying these genomic technologies, there's again some challenges here. So nurse bees and forager bees will feed themselves, right? So you can get double-stranded RNA in food to go directly to those nurses and foragers. But the nurse bees are basically acting as sort of the communal stomach for the rest of the colony. So they're the ones who provide the food to the larvae, the queen, and the drones. Right, so if you're feeding double-stranded RNA for an RNAi approach, it means that it has to go through the nurses before it gets passed to the other bees in the colony, and so that might reduce its efficacy. Right, so in this example here, it's a bee that's directly feeding off of the food, and this has some double-stranded RNA that's affecting varroa mites, for example, and then this causes the varroa to die. So this system works for nurses and foragers, but it would not work for the other bees in the colony. But there is an alternative approach, and so this is, as Rose was talking about, you can modify bacteria to produce double-stranded RNA in large quantities. And so, of course, honeybees, as with every organism, have a lot of microbes that live in their gut. And so you can modify one of these species and have it produce double-stranded RNA. And then that bacteria can be sort of stably producing this, and also it can be passed to other bees in the colony. So it can be sort of spread throughout the colony. There's some concern that this could also be spread to bees outside of the colony, and so that's a consideration. So Nancy Moran's work just published a really great series of studies doing this approach, and they used double-stranded RNA in bacteria that was against deformed wing virus, and bees that were treated with this bacteria and exposed to the virus lived longer than untreated bees. And they also made a version that had double-stranded RNA against varroa mites, which are sort of the worst parasites that US and European beekeepers have to deal with. And again, this was successful in reducing mite levels on these bees. So another challenge for applying these genomic technologies is that stressed workers tend to mature faster. So they go through this process of behavioral maturation more quickly. And again, then that means they're becoming precocious foragers who don't live as long and who don't bring in as much food. So if they're not bringing in as much food, you can't rear enough brood. And if they don't live as long, then they die off, and that will drive the younger bees to become precocious foragers earlier. And so this can unbalance the colony demographic structure so that the colony collapses, right? So you basically can't produce enough bees to replace the foragers that are dying because you're not bringing in enough food. And there's a really great review by Andy Barron sort of on this idea. So you would have to make sure that whatever modifications you're making are not somehow stressing the bees and leading to this accelerated behavioral maturation. So, all right, so let's now think about like, how could we maybe apply this technology to honeybees to make more resilient bees? And so one approach would be to say, let's boost the immune function of these bees so that they're more resistant to viruses and pathogens and other diseases. And so immune pathways in bees have been mapped out very well. So they're similar across insects, and you can see they have very complex immune systems. This was from a review from Alex McMenamin, who was a master's student in the lab. He is now a PhD student with Michelle Flanagan at University of Montana. And so this approach was used by Judy Chen a couple of years ago, where she focused on this gene called naked cuticle, which is an antagonist of immune signaling. So it means it suppresses immune signaling in insects. And she and her team had found that expression of this gene is increased by bees that are infected by Nosema, which is a microsporidian gut parasite. And so the thought was that Nosema was upregulating this gene to turn off the immune system so that the bee was not able to fight it off as well. And so the theory would be then if you knock out naked cuticle, then the immune system is active again, and then you would be better able to combat Nosema infections. And that is exactly what happened. So she knocked down naked with RNAi, and this caused an increase in expression of the immune genes, a decreased parasite load, and an increased lifespan. right? So this is great, right? So this sounds like this approach should work. So this, so can we just increase the immune function of bees and then have bees that are more resilient? And I would argue that there's these immune responses are very pleiotropic, which means that they're linked to many other molecular, physiological, or behavioral pathways and traits. So if you turn them on consistently, you might have some unintended consequences. And so for an example of that, a few years ago, Robert Paxton and I led a group that was funded by the German Center for Integrative Biodiversity Research, where we took all of our transcriptome studies. So these are all these studies where we looked at gene expression responses to bees that were infested with Varroa, Nosema, or viruses. And then we looked for common gene expression responses across all of these studies, or that were specific for particular parasites. And so what we found was that we found 344 differentially expressed genes that were consistently regulated across all of these studies. And then we also found some that were specific. And again, when we look at these, a lot of them are coming up in these immune pathways from bees, right? So it seems like the immune pathways are being affected. But we also saw a lot of other genes that were being affected that are not really part of the immune pathway. And so vitellogenin is one of them, P450s, which is involved in detoxification, and many metabolism genes. And so vitellogenin is particularly interesting because it is a marker and also a regulator of behavioral maturation. So its expression is much higher in nurses than in foragers. And if you reduce its expression, those bees become foragers faster, right? So potentially this response that we're seeing to immune stimulation could be some related to this kind of accelerated behavioral maturation that we see when bees are stressed or are sick. And then that could lead to overall colony level responses. And so there is sort of this theory that, when a host like a bee is fighting an infection, investing too much in the immune response may actually be the problem, right? So the immune response itself may be having a negative effect on the organism versus if they just sort of, don't accelerate this immune response, then they may live longer and they would be more tolerant. So this is something to consider at any rate if we were thinking about making these sort of high efficiency immune pathways. And again, this is because these stress workers might lead to an imbalance in the colony. The other thing that I wanted to point out is that immune function is linked to many other traits. And so this is work that was done by Claire Richoff. She was a postdoc in the lab and she's now an assistant professor at University of Kentucky. And so what she showed was that the social environment that bees experience as larvae influences their adult behavior and health. And so she screened for colonies that were highly aggressive or very docile, meaning they were not aggressive at all. And then she swapped frames of brood between those colonies. So bees were reared in a low or high aggressive colony background. And then she found that there were a lot of effects that were maintained in the adults of those bees. So bees that were reared in colonies that were more docile, so less aggressive, were more docile themselves. They were less resistant to pesticides. They had higher levels of Varroa. And when she looked at their expression patterns, they looked like immune stimulated bees. So again, these traits are not really separated and able to sort of be separated from each other. So in terms of broader ecological concerns, so one thing to consider is that if you sort of make your host have a better immune response, then you might select for pathogens and parasites that are more virulent and better able to deal with or outrun that immune response. So there's been some work that's been done by Andrew Reed and David Kennedy at Penn State looking at imperfect or leaky vaccines. And so if you have a leaky vaccine, it means it doesn't totally block transmission of that pathogen. And under those conditions, you actually select for pathogens that are more virulent, they go to higher titers faster, and they get transmitted more quickly, right? So this could be, again, a potential outcome of having a more immune efficient bee. There's also lots of other bee species, right? So if we were applying these technologies to something that the other bee species are also experiencing, right? So say we made a honeybee that was more resistant to pesticides, that doesn't really help the other bee species very much. And so we have to sort of consider this broader community when we're trying to think of these technologies. And then finally, you know, sometimes it's better, I think, to think about the drivers of these stressors that we might be trying to protect the bees from, rather than trying to make the bees more resilient to these drivers. So this was work that was done by Maggie Douglas that she started when she was a postdoc in the lab, and she's now an assistant professor at Dickinson College. And so she was mapping out insecticide use across the US over the last 20 years. And looking at the overall toxic load of those insecticides. And as you can see, the overall toxic load is increasing quite dramatically. This is almost entirely from neonicotinoids. And when we mapped out the crops and the spatial patterns of these of this use, this is pretty much coming almost entirely from seed treatments of soy and corn in the Midwest. And as you'll hear from John Tooker, I think on February 9th, these neonic seed treatments don't actually seem to improve the yield of these crops very much. And so the question is, why are we using these? And so rather than maybe making bees that are more resilient to these particular pesticides, maybe we should rethink the way that these are being used. So with that, I'd just like to sort of summarize and say this, these genetic technologies clearly can be applied to bees, and they can be used in ways to make them more resilient to common stressors. And again, like some of these stressors that honeybees deal with, like varroa and deformed wing virus, it would be great to have some alternative treatments for them because they're really problematic for honeybees. But there are aspects of bee biology and ecology that make it difficult to sort of deploy these approaches very easily or have them be effective in the long term. So with that, I'm happy to take any questions that you might have.
This seems like a pretty quiet week.
It is.
Christina, I have
Yeah.
a question.
Sure.
Did you apply these methods also to other bees? Can you tell us a little bit about that? To bumble or even solitary bees?
Yeah, so I think RNAi in general can be applied pretty readily to multiple species, and Rose can probably talk a bit more about this. But that that has been used in other species, and bumblebees for sure. And I'm not super up on the other species, but there's no, as long as you can feed it or inject it, it's pretty straightforward to do those studies. The CRISPR work, again, there's no reason that it could not be applied to other bees, but I think it's more of a challenge in just that you do have to have a lot of eggs that you inject, or in the case of the remote system, you have to have a female that's producing a lot of eggs so that you can sort of screen enough to be able to find your mutant. And so I'm not, it's been applied to honeybees successfully, also by Martin Bai, but I do not recall seeing it in bumblebees. But again, the technology should work. The limitation is more in the production of the enough eggs to screen.
Okay, well, we have a couple of questions that come through the chat. So in the interest of maintaining the recording in an understandable way, I'm just going to read that out loud. Marius put in a comment about Rose's talk. How can negative impacts on beneficial insects and organisms be forecasted and assessed?
Yeah, I think this is potentially is because when we choose for example, we choose a lethal genes, to target our interest pest. However, these genes potentially can share the sequence similarity to other organisms, which may be benefit, to the systems. Yeah, there are many studies, to design this double-strand array, based on your target sequence, but at the same time use other beneficial organisms sequence. Then they perform the similar biological experiment and check if they can cause the mortality in other organism. Yeah, so far there's a safe. Yeah, but as I mentioned, because the genome information is still not available for all the organisms, right? You can't 100% confirm it.
Okay, and another question. Is there any molecular method that can be used to unlink genes such as aggression and mite resistance?
Yes, so this is an interesting thing that I think a lot of beekeepers and researchers have thought about, because so the Africanized bees and the African bees are more resistant to varroa mites, but they're also more aggressive, and that makes it difficult to keep them as a beekeeper. And so there's been sort of this long interest, is there some way that we can, keep the mite resistance genes or the mite tolerance genes, but have gentle bees? And there is actually a population of gentle Africanized bees in Puerto Rico that Tagrul Garai is working with. And I'm not sure, I don't think they're more mite resistant anymore though. So if we knew, the molecular mechanisms that underlie, sort of this link between aggression and mite resistance, then you could target that with an RNAi approach or a CRISPR approach to modify that, to unlink that system if you could sort of go on the ends of the branches of that, right? So if it's something about the sensory biology and their ability to detect the mites and then perform the hygienic behavior, you might be able to modify that without sort of influencing these other sort of physiological pathways that might be linked to other things. So again, if we, it's sort of a chicken and an egg thing. So we have to use these genomic technologies to really understand the molecular mechanisms. And then once we know that, then potentially we can use them in a way to make bees that are going to be, more resistant to these stressors, but easy to manage.
Okay, and we'll take one more question, and then I will have our student teams and everybody else jump over to our private discussion group for the rest of the class. For Christina from Lauren Young, how widespread are these technologies being used for commercial hives currently?
They are not. So I know that there was work that was being done by a company that Logics, and then that company was, or Monsanto acquired that company, and so they were making an RNAi treatment against viruses, which they're calling Remabee. And I know that was in large scale testing, but as far as I know, nothing has been reported from that, and it's not available for beekeepers. So yeah, so I think this was a technology that there was investment in, sort of at this, large scale commercial level, but I do not believe it's come to market yet. And there's, again, I have not seen anything really reported about those studies.
All right, well, thank you to Rose and Christina for their talk, and to Erica and Marlene for introducing our speakers this week and coordinating with the rest of us to prepare for this. And I think we're good for the week. So thank you so much all for attending, and for the students, we'll see you in our discussion room very shortly.